Okay, can somebody confirm you can hear me?
I can hear you.
Thank you. Before we begin, are there any questions? All right, if there's no questions, we will begin. Let me share my screen. Should be able to see my screen now. We should be on chapter 8, where I'm not getting a link saying where we were. Oh, there it goes. Did we get to there? Did we start talking just about the operon? Anyone?
Yeah, I think that's where we were.
Okay, great. All right, today is May 23rd. We should be finishing chapter 8 today. I don't know if we'll make that, but we'll try. You had the quiz yesterday. There is no lab today. I will log in for the first 15 minutes to answer questions. So in the lab time, I'll only be there for the first 15 minutes. And I'm only there to answer questions. If you do not have a question, you do not need to come and log into the lab. You should be working on your unknown project. And let me remind you, this project will take most of you about 2 weeks to finish. So you need to get started right away if you hope to finish this project. Very few students are lucky and finish it within a week or less. I think there is one student who's finished it already, and they were very lucky. They just happened to ask the correct test, and it gave them the exact answer for what they were looking for. I think they asked for a confirmation test after that, but still, they only needed 2 additional tests or something like that to get the answer. Most students will need to ask more than that, and like I said, it'll take them about 2 weeks. A few students are going to be unlucky, and it will take at least 3 weeks for them to finish the project. You only have about 3 weeks left in this project. Any questions about the unknown project?
I have a question.
Yeah.
I had a labeling error on my first test submission. Will there be a penalty for that, or am I able to resubmit for a new test?
If I haven't sent you the test, you can re-request a test, and I won't look at the older data. I'll look at your newest data.
Okay, thank
Okay,
you.
so if I haven't sent it, you're fine. Just re-put the test in. I have not sent the test request today, but I did for yesterday. All right, so let's get to chapter 8, and hopefully we'll finish it, but I don't think we will. We're actually supposed to start chapter 14, but we'll see how far we get. So we had talked about an operon and mentioned an operon is sort of like a supergene, where the gene of interest is called a structural gene, and the operon includes the structural gene, perhaps other structural genes, and then controlling regions around that structural gene. We're going to talk about the lac operon, and it's really the only operon we're going to talk about. When we control gene expression, it can be controlled either by induction or by repression. The lac operon, which is the one we're going to talk about, is an inducible operon, which uses obviously induction. And that means that the gene is normally turned off, but when you want the gene turned on, the cell can turn it on. So it is inducible. We're not going to talk about a repressible operon, and you will not be quizzed on it. A repressible operon is a gene that is normally turned on, but when the cell wants to turn it off, it can turn it off. Any question about any of that? So the lac operon's the only operon we're going to talk about. It is the operon that controls the metabolism of the sugar lactose. Now, in E. coli, as well as all bacteria, and as far as I know, all archaea, but I'm not an expert on archaea, the lac operon in all of these prokaryotes has 3 structural genes, Z, Y, and A. The Z gene, or the first gene in the operon, the first structural gene in the operon, is the gene that codes for lactase. And it does have another name, which I've forgotten what its name is. Beta galactosidase. That's the same thing as lactase, the enzyme which digests the sugar lactose, breaking it down into glucose and galactose. Any question about that? The other structural genes in the lac operon are 2 genes involved in the metabolism of the sugar lactose. The Y gene is a gene that codes for the transport protein that moves the sugar lactose across the cell membrane. And then the A gene is involved in the conversion of the sugar lactose to the sugar glucose. So the A gene helps in the sugar of helps in the metabolism of lactose, and that is it helps convert when the Z gene splits the lactose into glucose and galactose. Glucose can be used because that's the base sugar, but galactose has to be converted to glucose, and the A gene helps in that conversion. Any question about that? So all of the genes in an operon, or I should say all of the structural genes in the operon, are related genes. In the case of the lac operon, all of the structural genes are involved in the metabolism of the sugar lactose. And as far as I know, that's true of all operons. They only have related genes. Any question about that? Meaning genes related in the same metabolism. So how the lac operon works is the RNA polymerase binds to the promoter and then moves down the DNA, moving across the operator, and then would transcribe the Z, the Y, and the A gene in the lac operon. And the terminator is at the end of the A gene, so the RNA polymerase would fall off. But in the case of the lac operon, this gene is normally turned off. How it is turned off is from the I gene. The I gene is a regulatory gene that controls the expression of the lac operon. Normally, in the case of regulatory genes controlling the genes that they regulate, they are not found anywhere near the genes they regulate. Meaning in E. coli, most of the regulatory genes are not found anywhere near the gene it operates. And in humans, the regulatory genes are usually found on a different chromosome than the structural gene that it regulates. Any question about any of that? But in the case of the lac operon, the structural gene is right next door to, not the structural gene, the regulatory gene is right next door to the structural gene that it controls. So on the lac operon, the regulatory gene is right near the lac operon. Now, let me state that this regulatory gene, which is given the letter I there, is not a part of the lac operon. It is its own operon, meaning that's the structural gene there, and above it, it would have a promoter. I don't know if it has an operator. I'm sure it has a terminator. So it does have regulatory regions. And so it is its own operon. You don't need to know that because we're only talking about the lac operon. But what the regulatory gene does is when it's transcribed, it makes a repressor protein. This repressor protein binds to the operator. When the repressor protein is bound to the operator, then the RNA polymerase that binds to the promoter tries to move down the DNA, and it's stopped by the repressor protein. That shuts down the expression of the lac operon, meaning the lactase gene and the permease gene and whatever that one was called, the something acetylase or something like that. I'll show you those names, but the point is the lac operon gets shut down when the repressor protein binds to the operator. And what it simply does is it blocks the RNA polymerase from moving down the DNA. And that's how the lac gene is normally turned off. The repressor protein is made, it binds to the operator, and then the Z, Y, and A genes are not transcribed. Any questions about any of that? All right. Well, you need to have some way to turn on the gene. And how that's done is there is an inducer which turns on the gene by inactivating the repressor protein. In the case of the lac operon, allolactose is the inducer. Allolactose is an isomer of the sugar lactose. One of them is the L form of the isomer. The other is the R form, meaning the left-handed and the right-handed. So they're the same sugar. They're just isomers of each other. And whenever you have the sugar lactose around, you'll have allolactose around. Any question about that? And the isomers, if you don't remember from Biology 160, whenever you have one isomer around, it will make the other one with time just because it's the same molecule. I don't remember the configuration for lactose, but it's something like C12H24 O12, something like that. And whenever you have one of the isomers around, given enough time, it will have some of the molecules convert to the other form. The point is, whenever lactose is around, allolactose will be also around. And whenever allolactose is around, it'll bind to the repressor protein. That will inactivate the repressor protein, so it will not bind to the operator. If it doesn't bind to the operator, then when the RNA polymerase binds to the promoter, it'll move through the operator, transcribe the Z gene, the Y gene, and the A gene. And that is how the lac operon gets turned on. In short, there's a few complications, but we'll talk about those. Any questions about that? All right. Well, here's something unusual. We never seen a messenger RNA like that. How many proteins are made from this messenger RNA? Boy, you guys really do not like to answer questions. Well, you can see it from the screen, so I'm not going to say. And in the case of the lac operon, more than one protein is made from this messenger RNA. In the case of bacteria. Now, in the case of humans, we do have a lac operon, but remember in the eukaryotes, they only have one structural gene in an operon. And so in the case of humans, we have a operon for the Z gene, a second operon for the Y gene, and a third operon for the A gene, because humans metabolize the sugar lactose as well, and we have to have the gene to make the enzyme lactase, which is also called beta galactosidase. They're the same term, mean the same thing. And permease is the gene that codes for bringing the sugar lactose across the cell membrane of the cell. So that greatly speeds up the entry of the sugar lactose into the cell as you make the permease gene or the transport protein, which brings the sugar galactose into the cell. And then transacetylase is involved in converting the sugar galactose to the sugar glucose, which you need in the metabolism of the sugar lactose. Convert it into glucose, and of course that's when the first part of the lactose is glucose, but you need to convert galactose also into glucose to metabolize the sugar. Any question about any of that? If not, let's watch a little movie on the lac operon, and then I'll come back and finish talking about the lac operon.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on two of the three proteins encoded by the lac operon: beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region.
That's the operator.
Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the control.
Okay, let me state that's the one part where this movie is not correct, and that is lactose does not bind to the repressor protein. Even allolactose, which is the real inducer, does not bind to the repressor protein when it is bound at the operator. It can bind to the repressor protein when the repressor protein is unbound and free. So the video is being a little bit oversimplified. I'll talk about that later when it's saying it inactivates the repressor protein by binding to it, and then that makes the repressor protein fall off the lac operon. Once the repressor protein is bound to the operator, allolactose cannot bind to it. And it's just because the binding changes the binding site that allolactose binds to the repressor protein, and you need to have a free protein to inactivate it.
molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
All right, any questions about that video? If not, let's go back to the lac operon. Let's see. So, what really happens to the repressor molecule is once it is bound to the operator, allolactose cannot bind to it. However, the binding of the repressor protein to the operator is not a permanent reaction, meaning it binds by a stoichiomic reaction, and that means the repressor protein sometimes falls off. If there's lots of repressor protein in the cell, then another repressor protein will bind to the operator and it will keep the lac operon shut down. Okay? However, if allolactose is around and it inactivates the repressor protein, then the repressor protein that's bound to the operator sooner or later will fall off. And once it falls off the operator, allolactose can then bind to it and inactivate it. And once it falls off, that will turn on the lac operon, meaning the polymerase, when it binds to the promoter, can just move down and transcribe the gene. And here's another question for you. What happens to that RNA polymerase when it binds to the promoter and tries moving down the DNA and gets stopped at the operator by the repressor protein? Is that RNA polymerase permanently kept there? Anyone? Anyone think the RNA polymerase is permanently kept there? Let's try and have a vote where you guys can raise your hands. I'll stop the share and then I'll tell you what the votes were. So raise your hand if you think the RNA polymerase is permanently kept there. All right, nobody raised your hand. So the rest of you, raise your hand if you think that the RNA polymerase will not be permanently kept there and it will eventually leave. 3 participants raised their hand there. What do you know? 4 participants raised your hand. All right, you 4, you're correct. What happens is the, let me share my screen again. The RNA polymerase is not permanently blocked. Once it goes here, the RNA polymerase binds to the DNA in a stoichiomic reaction, and every once in a while, it falls off the DNA. And so, yeah, it will bind there and then move to the operator and get stuck, but it's not stuck there forever. Every once in a while, it'll fall off, and then it'll bind to another promoter, either the same promoter or another promoter, so that RNA polymerase is not permanently stuck there. Any question about any of that? All right, let's talk a little bit about why, in the case of E. coli, would the gene be induced? Meaning, why is the gene, the lac operon, normally shut off in E. coli? What do you guys know about the sugar lactose? Where is the sugar lactose found? Where do you find the sugar lactose and what products?
Is it in dairy?
Yeah, it's in dairy. So how often would E. coli happen to find dairy products? And we're talking about E. coli not in a person's gut, like E. coli in the soil or E. coli on an apple. How often would E. coli see the sugar lactose? For most E. coli, it would not see the sugar lactose very often. The only exception would be if the E. coli happened to be on a dairy farm, then it might happen to find the sugar lactose. Or the E. coli is in somebody's gut, because obviously people can drink milk. I don't, but I'm lactose intolerant, so I don't drink milk. But there are many, at least Europeans, that do drink milk. So except for those E. coli, most E. coli would not see the sugar lactose, because lactose is not a common sugar. So that is why in E. coli, the lac operon is normally shut down. However, when E. coli does happen to be around the sugar lactose, then allolactose will be present, and allolactose will bind to the repressor protein, inactivating it, and that will sooner or later turn on the lac operon. Any question about any of that? And then when the lactose is all metabolized, there will be no more lactose around, so there'll be no more allolactose around. And that means the repressor protein will become active again. Once again, this is a stoichiomic reaction, and it will fall apart every once in a while. All right, I think that's it for the lac operon. Let me state that eukaryote operons are very similar to prokaryote operons, with one exception, and that is a eukaryote operon only has one structural gene in the operon. In a prokaryote operon, there may be one structural gene, or there could be several structural genes that are all related to each other in the same operon, such happens in the case of the lac operon. Another difference between E. coli operons and human operons or eukaryote operons is that eukaryote operons have more regulation than bacterial operons. All right, let's move on to mutation. Mutation is a change in the genetic material. Mutations may be neutral, beneficial, or harmful. Harmful is one where the mutation harms the individual that has the mutation. A mutagen is an agent that causes mutations, but some mutations can happen in the absence of a mutation. I mean, a mutagen, and that we call spontaneous mutations. These occur in the absence of a mutagen. They just are a mistake. Every time the DNA copies a nucleotide, there is a chance that the DNA polymerase could make a mistake. And that would be a spontaneous mutation, if that were to happen. there are different types of mutations. There is a base substitution, also called a point mutation, where one base pair is changed. And these can be a silent mutation, a missense mutation, or a nonsense mutation. So there are three different types of a base substitution. There's also a frame shift mutation where either a base is added, so the DNA only has 3 nucleotide bases, and then you add a base, and so now we have 4 nucleotide bases. Or you could subtract a base. That we call a frame shift mutation, and I'll talk a little bit more about why that's called a frame shift mutation in a little bit. And then there's a third type of mutation which your textbook does not discuss. And a macromutation is what it's called. This is where an entire region of a chromosome is added or it is deleted. I do not know why your textbook did not discuss a macromutation. An entire region of a chromosome, or maybe even an entire chromosome, is added or deleted. I should also warn you that the term macromutation could be used as I described, or it could be used as a mutation which causes several changes in an organism, meaning where more than one phenotype is changed. And that would be a regulatory gene, where when you change that regulatory gene, you change a whole bunch of other genes that are controlled by that regulatory gene. And we're not going to talk about that. And I'm just warning you that macromutation can be used in 2 different ways. We're using it as a large region of DNA, a part of the chromosome, or maybe the entire chromosome, is changed, added, or deleted. And actually, most of you know a macromutation. What is Down syndrome? Down syndrome is where an individual is born with an entire extra chromosome. I think it's chromosome 23. Is that correct? Anyone? Nobody's going to do it, so I'll look it up. I used to know this off the top of my head, but now I've forgotten. I think it's chromosome 23. Why didn't that go?
I think it's 21.
Is it 21? It could be 21. It's either 21 or 23.
Yeah.
Oh, there it goes. For some reason, my browser was doing that. Yep, it's 21. You're right. Thank you. All right, let's go back to where we were. So that's a macromutation you do know. Another macromutation is a hermaphrodite. Someone is born with chromosome XXY. That's another macromutation. Most macromutations where the entire chromosome is added or deleted will kill the embryo, or at least the fetus, so the individual is never born. But in the case of Down syndrome, they are born because the chromosome is not having an extra one. It's not lethal, but it does change the individual. Obviously, the XXY individuals are pretty much phenotypically normal, although they may be having traits of both male and female, but they are fairly phenotypically normal. You can also have a individual who's chromosome X, meaning they're not XX, or an individual who's chromosome XXX. And these individuals are born. And then the other one, which is XYY, they appear to be phenotypically normal, meaning we do not notice any difference between an XYY individual and an XY individual, other than the fact that the XYY individuals are a little bit more aggressive. Most other macromutations where the entire chromosome is either removed or deleted are actually lethal. Here's a picture of an individual. They were checking to see if this boy was a mutant. And as you can see, I have to blow it up maybe. The child has spoons that stick to him. And they were checking to see, is he a magneto? There, you can see the spoons are just sticking to him. Other people thought he might have just had sticky skin, and they were looking into him to see what was going on. And I'm afraid I never actually got the follow-up article discussing what they found when they studied him. But they were going to study him, and it's just a possibility the child was a mutant. All right, a base substitution is when a nucleotide gets inserted that's incorrect. And then when that DNA replicates, the T will then fix the mutation, meaning that while the nucleotide pair is GT, this won't bind correctly. And so the cell can always fix it, because it'll notice that there's a mistake there, and it can remove the T. It'll assume that the mistake is on the newly replicated chromosome, which is most of the time true. And then it'll remove the T and add a C. So it can repair that. But if the DNA replicates, this one will actually, come on, mouse. This one will replicate normally, but this one will not. And then that will fix the mutation. And in this case, this base substitution, where we change one nucleotide base by another, will change the amino acid. That is a base mutation. The DNA polymerase makes these mutations about 1 in every 10 to the 9th nucleotides that the DNA polymerase copies. Or what it does is it reads this strand as a template and inserts the correct base here. About 1 in 10 to the 9th bases that it adds, it makes a mistake. The base mutations can be a silent mutation. A silent mutation is where the DNA is changed, but the protein is not. What happens in a silent mutation is where you have the codon UUU, you can mutate the DNA, or in this case, the RNA, UUC, and it codes for the same amino acid. So that mutation would be a silent mutation. where the DNA is changed, but the amino acid is not. So there's no change in the protein. And that's a silent or neutral mutation. Any question about that? You can also have a missense mutation where one change in the DNA base is changed, and it does result in a change in an amino acid. For example, we have the codon CCG, and when the messenger RNA is made, that codes for the amino acid glycine. And if we change the CCG to TCG, that will change the amino acid from glycine to serine, creating a missense mutation. You can find a missense mutation in sickle cell anemia patients. Another missense mutation that happened 10,000 years ago in the human population is the brown eye gene got changed in a missense mutation, resulting in the blue-eyed allele, the blue-eye gene. And then humans, which before that point only had brownish eyes, now had brown eyes and bluish eyes. Any question about that? And they know this happened in Northern Europe about 10,000 years ago. It's a simply a one missense mutation, meaning one base change in the DNA that resulted in a one amino acid change in the protein, resulting in the blue eyes. Any question about that? You can also have a nonsense mutation where the DNA is changed in one base, but instead of changing an amino acid, that codon now changes to a stop codon. Let me blow this slide up. So here we have, I wish they had grouped these so I can see it a little better. Let's see, it's going to be ATC. So here we have TTC, and we have a base substitution where that T here is converted into an A. And that, instead of coding for the amino acid phenylalanine or maybe lysine. Yeah, it's lysine. So sorry, it's TTC. Instead of coding for the amino acid lysine, it codes for the stop codon. Now, when the protein is made, the amino acids before the stop codon will be the same, but after the stop codon, you have no more amino acids in the protein. That will result in a truncated protein. That protein usually does not function. And if it does function, it is definitely going to function at less than optimal rate because a large part of the protein is missing. Oops. Back. An example of a nonsense mutation is a mutation in some cystic fibrosis patients where they have the DNA changed, and instead of it creating a amino acid or codon that codes for an amino acid, the codon is changed to a stop codon, resulting in a truncated protein. and that protein doesn't function correctly, resulting in cystic fibrosis. Any question about any of that? All right, let's talk about a frame shift mutation. A frame shift mutation is where you insert one or more bases into the DNA, or you delete one or more bases in the DNA. Let me blow this up so I can see it a little better. So here we have the codon AAA, coding for the amino acid phenylalanine, and we remove 1A. Now the codon is only AA, and you need to have 3 bases to have a codon. And so what the codon does is it just takes the next codon or the next nucleotide. So instead of being AAA and then CCG, it becomes AA and then takes the next nucleotide, which is a C. And then the next codon, instead of being CCG, which is right here, it'll start here and go CGA. And you can see why this is called a frame shift mutation, because once you change the nucleotides right here by adding one or deleting one, all of the other codons after that are changed. And that's why it's called the frame shift mutation. Now, let me state there is one exception to a frame shift mutation, and that is if you subtract 2 nucleotides, that is a frame shift mutation. But if you subtract all 3 nucleotides, or a multiple of 3, then that is not a frame shift mutation. Because if you were to remove all 3 of these nucleotides, you don't have a frame shift mutation. What you do is you have a protein minus 1 amino acid, phenylalanine, but all of the amino acids afterwards will be the same amino acids. So that would be a missense mutation. You don't need to know that, but my point is that if you remove or add a multiple of 3, meaning 3, 6, 9, whatever, you don't have a frame shift mutation. Any question about that? Because the frame shift mutation is after that mutation, the entire protein is changed after that. Because the reading frame of the codons has changed. Some Tay-Sachs patients are examples of a frame shift mutation, where a nucleotide is either added or inserted, or a multiple of some number other than a multiple of 3. Any question about any of that? All right. As I stated, there are 2 causes of mutation. The mutation can be caused spontaneously, where when the DNA is just replicated, a mistake gets made, and that we call a spontaneous mutation. The other cause of a mutation is a mutagen. There are 2 types of mutagens. There's radiation, which can cause mutations, and there are chemical mutagens, which can also cause mutations. There are 2 types of radiation which can cause mutations. There's ionizing radiation. Ionizing radiation, like X-rays and gamma rays, have a lot of energy with them. And when they hit a molecule or an atom, they give the electron enough energy so that that electron leaves the atom or the molecule. And that will cause an ion here, because if this electron leaves, then this atom or molecule will have a positive charge because the electron has a negative charge. Any question about that? When you have ions forming, the ions can react with DNA and then mutate the DNA. That's ionizing radiation. You can also have a mutation caused by non-ionizing radiation. There's only one type of non-ionizing radiation that causes mutations, and that's UV light or UV radiation. UV light and UV radiation mean the same thing. Now, regular light that we see with is a form of non-ionizing radiation, and it does not cause mutations. Only UV radiation does. UV radiation causes a mutation when the UV light hits the DNA. If there are two thymines next to each other, the UV light can hit the thymines and then cause them to form two covalent bonds between them. And that's called the thymine dimer. A thymine dimer causes a mistake in the DNA because the thymine dimer, let's see if I can blow this up a little bit, as the thymines covalently bond to each other, which never happens except in the thymine dimer. And these two thymines do not hydrogen bonds to the adenine because they're binding to each other. This is a covalent bond, by the way, not a hydrogen bond. If the cell does not repair that, it will be a mutation. And we're not going to talk about how the cell can repair that, but this thymine dimer stops DNA replication, meaning the DNA polymerase will move down the DNA and then just stop at the thymine dimer because it doesn't know what to do with the thymine dimer. And it also stops transcription. So the RNA polymerase moving down the DNA doesn't know what to do with the thymine dimer, and so it just stops. All right, so that's how the two forms of radiation, ionizing radiation and non-ionizing radiation or UV light, cause mutations in the DNA. You can also have a chemical mutagen. We're only going to talk about nitrous acid as a chemical mutagen, but other chemical mutagens operate similar to the way nitrous acid operates as a mutagen. When nitrous acid binds to the DNA, it can change this adenine so that it's still adenine, but it's a modified adenine, and the adenine does not work as an adenine. And so when the DNA polymerase reads this template, instead of putting a T over here, because it sees an adenine, it won't see the adenine. It'll think it's seeing a C. And then it'll put, excuse me, it's thinking it's going to see a G. I didn't read that right. It's going to see this as a G, even though it's really an adenine, but it's a chemically modified adenine. And the DNA polymerase will read it as a G and then put a cytosine next to it. And then when this replicates, you fix the mutation. That DNA will replicate correctly, but this one will not. That's how nitrous acid acts as a mutagen. It actually binds to the DNA and then changes the DNA so that when the DNA is replicated, it doesn't replicate correctly. Instead of getting the GCT will get GCC. Any question about any of that? All right, I've already mentioned that spontaneous mutation occurs at one base being mutated in 10 to the 9th replicated bases. Because each gene has about 1,000 nucleotides, at least in E. coli, that means that the frequency of a mutation per gene is about 1 in 10 to the 6, or 1 in a million genes will be mutated. Now, that's not very much, is it? 1 in a million? But if you look at the human population, which is something like close to 8 billion, almost 8 billion, when children are born, 1 in a million will be a mutant. And that means mutant children are born someplace in the world almost every day. And this is just from spontaneous mutation. Mutagens increase the mutation rate of spontaneous mutation so that the mutation will be, instead of 1 in 10 to the 6 nucleotides, it'll be from 1 in 10 to the 5th down to 1 in 10 to the 3rd, meaning down to, instead of 1 in a million, down to as low as 1 in 1,000, or maybe up to 1 in 100,000. That will increase the mutation rate 10 to 1,000 times. Any question about any of that? Identifying mutants. You can test for mutants by a positive selection or a direct selection, where you can simply detect the mutant cells because they grow or appear differently. For example, if this were to go walking down the street, everyone would look at it and say, yep, there goes a mutant, because it looks different. But you can also identify a mutant by negative or indirect selection, where you can detect the mutant cells because they do not grow. So the cells look the same, but they actually do not grow. And let me talk a little bit more about that one. I actually did this when I was an undergraduate, and I was looking for mutations. And what you do is you get a colony where you have colonies of yeast or colonies of bacteria, and you have the colonies isolated. And then you get a velvet stamp, and you put the velvet stamp on the plate. That will transfer the cells from the colonies onto the velvet stamp. And then you stamp fresh plates with the stamp, so that this colony here comes from here. This colony here comes from here. And that colony will be from there. So there, and then there or there. Any question about that? So what you do is you replica plate, 2 plates, one containing the medium with histidine and another containing a media without that amino acid. And then you look for colonies that grow on the plate with the amino acid histidine. There's one there. And do not grow on the plate that's lacking that amino acid. So this is a mutant that requires the presence of the amino acid histidine, and the mutation is one where the cells cannot make that amino acid. And so it must be supplied by the media. Otherwise, the cell will not grow. That is negative selection for a mutation. Any question about any of that? Like I said, I actually did that as an undergraduate. All right. let's move on from mutation and then talk a little bit about genetic recombination. Recombination is the exchange of genes between two DNA molecules. Recombination usually occurs by crossing over, where two DNA molecules come together, usually the mother's chromosome and the father's chromosome, and then they cross over like that, so they physically cross over, and then they break right here and exchange pieces, where now the father's chromosome, which is the lowercase letters, has E, F, and G, lowercase, but then it has the uppercase H and the uppercase I from the mother's chromosome. Crossing over happens very frequently in both mitosis and meiosis of eukaryote cells. And the reason is thought that eukaryotes just want to shuffle the genes up, make new genetic combinations. And so crossing over happens very frequently in both mitosis and in meiosis. I will ask, how are recombination and crossing over differ? But I'm sure nobody will answer. So let me go ahead and tell you this one. Recombination is a more general phenomena where simply two genes are exchanged. Crossing over is the physical crossing over the chromosomes, and then the chromosomes change pieces. An example of recombination that is not involving crossing over would be, let's say the lowercase g gene were to leave this chromosome and go to the B chromosome, but there would not be an exchange of anything from B moving to chromosome A. That would be in a case of recombination, but not a crossing over event. Any question about any of that? So recombination is a little more general than crossing over. But usually recombination occurs by crossing over. All right, let's talk a little bit about vertical gene transfer. This occurs during reproduction when the genetic information moves between generations of cells, so that the genetic information moves from the parent cell to the offspring cell. When we're talking about humans, the genetic information moves from two parents to one offspring. That differs from horizontal gene transfer, where you have a transfer of genes between cells of the same generation, meaning DNA leaves the donor cell and enters a recipient cell. There are three different ways where horizontal gene transfer can occur: by transformation, conjugation, and transduction. Transformation is where DNA leaves the cell and then usually is naked DNA in the environment, and then that naked DNA can be taken up by a recipient cell. What happens is that DNA, you don't really need to know this, but that DNA will be taken up by that cell, and then there will be recombination between the DNA fragment that the cell took up and the chromosome. And then that chromosome contains a new gene, the lowercase a gene, which came from the DNA taken up by the cell. That can give this cell a new phenotype. For example, let's assume that this cell is sensitive to an antibiotic. And we'll just say penicillin G, but it could be any antibiotic. And then this lowercase a gene happens to encode for penicillinase, making the cell not sensitive to penicillin. and that lowercase a gene can get inside the cell and then recombine with the chromosome. And now that lowercase a gene is in the chromosome. And this cell will now be penicillin resistant, where that cell beforehand was penicillin sensitive. Any question about any of that? Here's a picture of transformation. The cell sees some DNA out there and then consumes the DNA. Obviously, it doesn't eat the DNA, or it brings the DNA into the cell. And then the DNA starts recombining with the chromosome, giving the cell indigestion. And then the gene is expressed on this piece of DNA. It can then change the phenotype of this cell from the newly gained DNA from transformation. Any question about any of that? All right, let's talk about conjugation. You can have conjugation occurring in one of two ways. In gram-negative bacteria, and as far as I know, only prokaryotes can do this, generally bacteria, that conjugation can happen by a sex pilus. The sex pilus is this long tube coming out of this cell. The sex pilus literally goes out and then tries to skewer an F- cell. This is an F+ cell. And we'll talk a little bit more about that. And then the chromosome from this cell, which is the F plasmid, what's called F+, can replicate and then send a copy through the sex pilus, giving this F- cell plasmid DNA. And that's sexual conjugation in a gram-negative cell. Sexual conjugation can happen in a gram-positive cell as well by a mating bridge. What happens is two cells come together and the cell wall where they touch happens to disappear, and then the cell membrane on this cell confused with the cell membrane on that cell. And when that happens, they can then have the cell membrane reanneal. So let me blow this up if I can. Where the cell membrane, instead of coming across like that, can anneal with the cell membrane here. And now the cell membrane runs this way, around this cell, around here, and then that way. So these two cells are literally connected with the cell membrane. And in addition, the cytoplasm of this cell is connected to the cytoplasm of this cell. That can allow DNA to move from one cell into the other. And that we call a mating bridge, which is sexual conjugation in gram-positive cells. So let's talk a little bit about sexual conjugation a little more in gram-negative cells, where the sex pilus, as I stated, literally from the F+ cell goes out and skewers the F- cell. Let me back up a little bit. And that's better there because it's actually showing you this cell is going out, and then the sex pilus literally skewers that cell. That sex pilus can then act as a winch to bring the two cells closer together. It doesn't need to, but it does do that. And the reason why it doesn't need to is this sex pilus is hollow, and that will allow the F plasmid to transfer from this cell through the hollow sex pilus into that cell. But it can bring the cells closer together, like here, and then the F plasma, what it does is it replicates and then sends a copy of itself across the sex pilus into the F- cell. This F- cell now has the F plasmid in it. And what the F plasmid does is it codes for the production of the sex pilus. So this F minus cell, when it gains the F plasmid, will start to grow a sex pilus. And then this formerly F minus cell can then go out once it has a sex pilus and skewer an F minus cell and then replicate its plasmid and send it across the sex pilus, meaning this formerly F minus cell is now an F plus cell. And that's sexual conjugation. Any questions about sexual conjugation? All right, let's talk about transduction. Transduction is the third way where genes that are existing in the population can spread from one place in the population to another. And obviously that's the way transformation and sexual conjugation happen, but transduction happens a little differently. And transduction uses a virus. What normally happens is, we haven't talked about viruses yet, but when we do, we'll mention that this virus here will bind to this cell, and the virus will insert its genome into the cell. And then the viral genome will want to call the shots in the cell. An easy way to call the shots in the cell is by the virus encoding an enzyme which digests up the cellular chromosome. When the cellular chromosome is all in pieces, that means the viral genome will be calling the shots in the cell, not the cellular genome. What the virus will do is it will take over the machinery of this cell and then replicate the virus, replicating the viral genome and then replicating the viral protein coat, also called the capsid. And then the viral genome will be inserted inside the protein coat. I should mention the protein coat of a virus can self-assemble. So we have the protein coat and then the genome is inserted. And then the virus will lyse this cell. That'll be the last thing it does. And then it will leave that cell once it gets lysed and then can go on and infect other cells. But viruses can make a mistake, and they make lots of offspring, so they aren't terribly worried with a mistake. And that is right here, we have the viral genome added to the virus. And so these viruses will replicate normally, meaning they'll infect the cell, take over the machinery of the cell, use the cell to replicate more viruses. But this virus has a mistake. Instead of getting in the reddish purple viral genome, this virus got a piece of the digested chromosome of this cell. Now let's follow this virus. It will infect another cell and then act normally by injecting its genome into the cell. But this isn't a viral genome. It's part of the genome of this cell. That transduced DNA can then recombine with the chromosome and then be expressed. Once again, that's very important for us because if this cell was antibiotic resistant, and then this DNA piece happens to contain the gene for that antibiotic resistance, and then this DNA were to combine with this chromosome, this recipient cell will now be expressing a gene from this cell, which gives the cell antibiotic resistance. And that's transduction, where existing genes in the population are spread around from one cell to another by a virus. Transduction occurs frequently in bacteria. Let me show you that now. We mentioned spontaneous mutation happens one time per 10 to the 6. Let's see if I can get that. Or for every gene, if you replicate 1,000,000 of them, you will get one spontaneous mutation. Let's just go there. Oops, I'm now on that. Let me get rid of that. There we go. Well, we can have not a mutation, but transformation. Transformation happens about 10 times more frequently than spontaneous mutation. And the same with, oops, got that too. I didn't mean to get all of that, meant to. Only get the latter part. Let's do that twice. Oops. Conjugation. And transduction. So transformation, conjugation, and transduction happen about 10 times more frequently than spontaneous mutation. This is a concern because if you're looking at maybe a case of antibiotic resistance, it may not have occurred from spontaneous mutation. In fact, it's 10 times more likely that it occurred from transformation, conjugation, or transduction. All transformation, conjugation, and transduction do is take pre-existing genetic information and then spread it around in the population. Any question about any of that? Humans actually have a form of transformation, and we call it sexual reproduction. We are a sexually reproducing species, and so we do have the genetic information mix between the mother and the father to give rise to the offspring. We do not have conjugation, as far as I know. I've never heard of anyone suggesting that eukaryote cells, let alone human cells, have conjugation. Transduction probably happens in the cells of your nose and maybe the cells of your throat all the time, where those cells are picking up DNA from another person. However, that doesn't change your germline. And so transduction is not very important in human evolution because usually transduction does not change DNA in the germline. Any question about any of that? All right, let's close this. Back to where we were. And say it's actually time to quit now. So I'll continue here next time. Are there any questions? I will be in the lab, but only for the first 15 minutes. After that time, I will log out. Last chance for any questions? All right, I'll see you.